So just a few more slides here to wrap up our discussion on an overview of metabolism. And so we've been very carbohydrate focused, and so we've been thinking about these pathways here that we have for carbohydrate metabolism, which are glycolysis to metabolize glucose to pyruvate. We've got the pyruvate dehydrogenase reaction here that converts pyruvate into acetyl-CoA. We commit acetyl-CoA to the citric acid cycle, and then all of these reduced nucleotides, these reduced cofactors, go on to the electron transport chain. At the end of the day, the magic number I want you to remember is 32, and we'll learn much more next semester about why it's not 38. If you're hearing 38, that's probably an old number, and it's based on old kind of conversion factors for how many ATP each NADH and FADH2 are worth. So 32 is kind of the magic number I want you to remember for the energy equivalent in one glucose. So again, metabolism of glucose fully to CO2 and H2O yields 32 ATP of energy. So we've mentioned the cellular locations that we have of things. But again, up here in purple is really carbohydrate specific, but once we get down to pyruvate and then specifically acetyl-CoA, we can start considering the metabolism of other fuels. And so those are the last couple of slides I have to go through here. So we want to think about fat metabolism, and remember that when we're talking about fats, we're really talking about triglycerides, and triglycerides really are metabolized to give us fatty acids that can then be further oxidized. So that process is called beta oxidation. And what I want you to think about when you think about beta oxidation is imagine these fatty acids that are here. And again, each of these orange spheres represents a carbon atom in this case. And what we're going to have, imagine if you ever had those little like pop beads as a kid to kind of string them all together. What we're going to be really doing with beta oxidation is snipping two of these off in one round of beta oxidation. So it's really interesting to think about. We don't chew in from the end like we do with glycogen metabolism, for example. We snip off these two carbon atoms that are kind of shown in gray here. And we're not going to learn the details of this process, but I do want you to remember the number of steps and where it occurs. So again, fat metabolism, beta oxidation of fatty acids, is 4 reactions, and it occurs in the mitochondrion. So again, the only process we've really learned about that occurs in the cytosol is glycolysis. Everything else occurs in the mitochondrion. Again, this is an aerobic process. This will not happen unless there's sufficient oxygen around. And so a 18 carbon fatty acid will generate 9 molecules of acetyl-CoA, and again, those are metabolized in the citric acid cycle, and then a large amount of this alternative currency, which is handled in the electron transport chain. So again, here is kind of a summary of these 4 steps. It has to do with an oxidation step, hydration step, oxidation, and cleavage. So I want you to kind of remember those sort of details. It's oxidation, hydration, oxidation, cleavage. Oxidation, hydration, oxidation, cleavage. And kind of going through that cycle is how we kind of fully modify and oxidize these carbon atoms that then are eliminated as acetyl-CoA, and then that goes to the citric acid cycle. So not numbers that you need to remember, because fatty acids, unlike carbohydrates, are variable in the number of carbon atoms they have, but what we can see is we get a number that's much larger than we see for glycolysis. So a couple of things to think about. Problems with fats, right? Your body prefers to run on carbohydrates. So again, that's a problem though if you're a diabetic, because you either lack that insulin signal if you're a type 1 diabetic, or you don't listen to the signal if you're a type 2 diabetic that allows you to bring glucose into the cells. So if you haven't eaten in a while, if you've tapped out your glycogen reserves, your body needs to have some way of kind of handling this. So in the absence of any carbohydrates you can kind of pull on in your body, your body's going to continue to break down fat to produce ATP. Again, that's the basis of how all diets work, is you tap into and you literally cannibalize your reserves of energy. Again, you can also break down proteins to make into glucose. All right, you need to maintain, a blood glucose level that's normal. So if you're not consuming any carbohydrates and you have tapped out your glycogen reserves, your body will convert proteins into glucose. And this is a really, really, really important point, so I'm going to say it a couple of times here. If you need to make glucose because you are not eating food and you've tapped out your glycogen reserves, How your body makes it is it breaks down proteins and uses specific amino acids that it can convert into glucose. Fats cannot be converted into glucose. I'm going to say that 3 times because it's super, super important. Fats cannot be made into glucose, but fats can pay the cost to convert other things into glucose. So fats cannot be made into glucose. But again, breaking down fat to this acetyl-CoA allows us to generate ketone bodies, and ketone bodies are a water-soluble fuel that we can generate from fat. So if we look a little bit at ketone bodies here, so again, when we go through that beta oxidation process and we get acetyl-CoA molecules, those acetyl-CoA molecules can be coupled together to make a 4-carbon molecule called acetoacetate. We can decarboxylate it to make acetone, or we can undergo a reduction reaction to make beta hydroxybutyrate. These green molecules that we have shown here are what we call ketone bodies. So they are a water-soluble kind of fat-derived fuel. And when you are in kind of a fasting state where you don't have glucose, you've got lower glucose levels, you haven't eaten, and you've tapped out your glycogen reserves, fat provides not only energy to convert other things into glucose, because you still need to maintain, that healthy level of blood sugar, but it also converts some of those fats into these green ketone bodies, which you can again circulate. They're very hydrophilic. You can circulate them in your bloodstream, and your brain especially will utilize ketone bodies as fuel. So we'll spend a lot more time talking about this next semester, but this state of ketosis is kind of where you are subsisting largely on fats as fuel. And it can be a dangerous situation if you get into what we call ketoacidosis, where you have too high a concentration of these ketone bodies and you elevate the concentration of protons, so you decrease your pH. You can cause this state of metabolic acidosis, and that's dangerous. But kind of existing in this ketotic state can be really important for diabetics. It can be really important for weight loss. So we'll talk a lot more about that next semester as we kind of learn how some diets work. And again, we talked a little bit about this. The state of ketoacidosis can also produce significant amounts of acetone, in being very volatile and very sweet smelling, can kind of indicate that somebody might be in this life-threatening state of a metabolic acidosis. All right, last little thing here is on protein metabolism. One of the things I want you to remember is the main purpose of consuming proteins is to break down those proteins into their essential amino acids so that you can rebuild them into your own proteins. So proteins are not necessarily meant to be metabolized as fuel, but when do we break them down? Well, basically, if there aren't any carbohydrates or lipids to use for fuel, we'll need to break them down. The other time that we will metabolize amino acids is if we have too many, beyond what's needed to make your own proteins. And again, there's a kind of a rule of thought for some diets that indicate that proteins should not comprise more than 10% of your daily caloric intake. And that's because you might not need more than that to fashion into the proteins you need in your body. So here's kind of the brief thing that we want to think about. We've already talked about peptide hydrolysis to give us these amino acids. Remember, amino acids are very water soluble, so this is kind of a hydrophilic circulating fuel. But there's things that we need to think about because we have a new atom to be concerned about with amino acid metabolism. We have nitrogen. So one of the things that we have to think about is kind of separating these into this part here, which we refer to as the alpha keto acid part. And then we're going to have the amine part. So we'll get into the details of how this happens next semester. But at the end of the day, this nitrogen for mammalian systems is converted into urea, and then urea is excreted largely in the urine. So we'll talk about the process for how you kind of snip off this nitrogen. And then once you have these carbon skeletons, these alpha keto acid skeletons, basically those will feed somewhere into the metabolic pathways that we've learned already. They'll feed somewhere into the citric acid cycle. They'll feed somewhere in along that pathway. So lots of details here. This is not anything that you need to know. But again, all of these skeletons, as you can see, feed into different parts of the cycles that we've already learned about, whether it's the citric acid cycle or it's kind of feeding in as pyruvate and so forth. But there are some amino acids that we can use via gluconeogenesis to make into glucose. So oxaloacetate is an important precursor in gluconeogenesis to make glucose. So we'll talk about this in detail next semester. So all I really need you to know is that once you snip off the nitrogen, the carbon skeletons that are left somehow need to be fully metabolized so that we can squeeze all the energy out of them. So that kind of just wraps up everything that we need to think about for metabolism of other fuels besides glucose. So again, we've got fats and then we've got proteins.